Hello everyone. Today's topic is Dige and toga. This video I will only be explaining the basic principle and the concept of Dige. We will see toga in the next video. Before we can understand what is the process of Dige, we need to understand the basic concept or the basic underlying principle behind the actual process. So let us understand the basics first. We have already studied what is one-dimensional gel electrophoresis. What you have done in the lab, that is your agarose gel electrophoresis or the SDS page, both those electrophoresis mechanism, it is a one-dimensional gel electrophoresis mechanism wherein the proteins are separated based on the molecular mass. This method is very easy to use for simple samples where the number of proteins are very few and they can be easily separated. The only drawback in this technique is this one is based only on one property, naming molecular mass. I can actually see Mugilan thinking, Mugilan's mind, thinking that ma'am, you have already told us that the separation is based on charge and the weight, that is the molecular mass. Then how come now you're saying it is only based on molecular mass? Charge, what we have understood, is used for movement of the protein, the direction, that's all. But the actual principle for separation is molecular mass. The separation of protein or DNA for that matter is based on the molecular mass. So let us see how the process takes place. Now, as I told you, one-dimensional, very simple method, very easy to use, but can be used with samples where the sample is a very simple sample, not many proteins are present, and they can be easily separated. To overcome this drawback, we use another type of electrophoresis system which is called as 2D page or two-dimensional gel electrophoresis mechanism. Now this is an advanced version of one-dimensional electrophoresis system wherein the electrophoresis is used for two important things. One is separation of the molecules and second, identification of the molecule. In this case we are talking about proteins, whichever protein is present in the biological sample. The basic principle is the same as that of one-dimensional gel electrophoresis system, except that here in 2D page the separation of protein is based on two concepts. The first one is isoelectric point and the second one is molecular mass. That means this in two-dimensional gel electrophoresis system, we carry out electrophoresis in two steps. In the first step, the separation is based on isoelectric point of the proteins which are present in the given sample. And in the second step of separation, the proteins are separated based on their molecular mass like we do in any SDS page. So this is the basic principle of two-dimensional electrophoresis system. Now the question is why we need two-dimensional page. Why not just go with a simple one-dimensional page? The reason is when you have too many complex proteins present in a mixture, then separation using just a single dimension, that is only molecular mass, may not give you a very clear picture. It will separate the proteins, but the separation will be very crude. Samples or proteins which are very close to each other in their molecular mass. For example, a protein is around 46.8 Dalton and the other one is around say 47 Dalton. If you do a normal SDS page, which is a one-dimensional electrophoresis, you will not be able to separate, you will not be getting two separate bands for these two proteins. Both of them will be will not get separated very clearly and you will see them as a single band. Hence, to overcome the problem of separation of proteins in a complex mixture, we use two separate dimensions which are run perpendicular to one another. Please understand, perpendicular to one another. To make you understand clearly, The first, for example, first run, if you are running it in this direction, the second separation will be in this direction. That is why we say the two separations are done perpendicular to each other, and each one is based on a different property. This one is based on, as I told you, the isoelectric point. That is why IEF. IEF stands for isoelectric focusing or isoelectric point. And this one will be separated based on the molecular mass. So this is called as IEF, and this one is called as your normal SDS page. Right? Okay. Now let us see how the process takes place. So here we require two important things. One, we need IPG strips. IPG strips are, I'll just show you, they are like this, thin strips, okay, like your ice cream stick, something like that. So these thin strips, on these thin strips the protein sample is placed here like this, and electrophoresis is like this. We have many strips. So imagine we are keeping many strips, and each strip we have loaded with one protein, the mixture of protein sample that we have. This is probably, first one is a separate sample, second one is from a separate sample, and the third one is from a separate sample. Let us assume this is 1, this is 2, and this is 3. The first one, let us assume, is from a normal person, protein collected from a normal person. Second one is second and third can be assumed as sample collected from patients, for example. These strips are called as IPG strips, and these IPG stands for immobilized pH gradient gels. These strips will have gel loaded on it. They are ready-made strips made up of thin plastic on which the gel has been casted. Imagine, like you have already done silica, you have already prepared silica gel. Silica gel, you prepare it on glass slide. Similarly, here on a plastic, a thin layer of gel solution is poured, and the gel solution is poured in such a way that it forms a pH gradient. This pH gradient is what helps us in separating the protein based on the isoelectric point. And this IPG, they are basically acrylamide gel matrix copolymerized with the pH gradient, which results in complete stable gradients. Let us see how the 2D page is performed. 4 steps. First one is sample preparation. Second one is to run the first dimension, which is called as IEF, isoelectric focusing. Second one is to run, next step is to run the second dimension, which is the normal SDS page gel. And last is visualization and analyzing the samples. So let us see one by one. The first step is preparation of sample. When I say preparation of sample, we are talking about isolating the protein sample from the cell, purifying the protein, and getting the correct sample with the correct pH and all that. So that is the sample preparation. What you are seeing here, this is IEF gel, and this one is the second SDS page gel. Let us see how each one is done. This is the IEF gel. So you can see here, this blue color, the bottom shield that you are seeing, that is, we have it in our lab. Next time when we meet, I'll show you how it is done. So here we have the platform on which 3 different strips have been kept, IPG strips have been kept, and you can see the sample is placed on one corner, and there is a pH gradient, pH 3 on the left to pH 10 on the right. So proteins, when you apply the electric current, they start migrating. So proteins have been placed here, here, and here, right? And they start migrating from negative to positive, and the migration will be based on their isoelectric point. Each protein has a different isoelectric point. Once each strip has been run and proteins have been separated based on the isoelectric point. Now here you can see, just look at this spot. There are two different proteins. Both of them are lying at the same spot. Normally, if you do SDS page, you will think that in this spot, when you see it as a band, it will be a single band. You will think there is only one protein. But actually, you can see how many proteins? 1, 2, and there is very close by, there is another protein, which is the third protein. So this will never be resolved in a normal SDS page gel. Similarly, here also you can see the same thing. So now let us see how it is resolved. So now we have the thin strip. That thin strip is now placed, you can see this is the thin strip. This is our SDS page normal. So here you have the stacking gel, here you have the separating gel, and on top what we have done is we have placed horizontally the thin strip, in the first step or in the first dimension gel that we ran, that strip is placed on top of the agarose gel. Not the agarose gel, sorry, the SDS page gel. Once it is placed, so this here from the top is now, after running, we are going to place it here. Once it is placed, we run a normal SDS page. Always we have to remember that the strip that we are placing on top should be in contact with the separate stacking gel, so that when the buffer runs from top, the whatever proteins were separated in one direction, I'll show you the direction. Initially the proteins were separated in this direction, right? Now when we place this horizontally on top of the SDS page, the proteins are now going to be separated in perpendicular direction. So whatever protein was present here, you can see is now slowly getting separated, and now whichever was forming a single spot, now you can see they are separated as individual bands. So this helps us in separating the proteins which are not very different from each other molecular weight wise, can be separated based on their isoelectric point and their molecular weight using two-dimensional gel electrophoresis. Till here I hope you have understood. So after that, to visualize and analyze. So this is how you can see here image. What we did in our lab was Kumasi brilliant blue staining. That is why you saw blue color bands. In this image they have done silver staining, and in silver staining the bands are not visualized as blue, but they are visualized as brown color. So, and all these spots that you are seeing on the left hand side, what you are seeing, this is a molecular weight marker, and all these are the spots that have been resolved. So this was supposed to be a single spot on the first dimensional gel, which has now got resolved into multiple spots. You can see how many spots we have got now after running a second dimension. So this is how the image is analyzed. You can see from a single spot you can identify multiple proteins. But then here we had certain difficulties or certain disadvantages. One, spot number, that is every cell, we know that every cell, it contains many genes and each gene will produce many products. So based on that, we have around 10,000 to 150,000 gene products in a cell, and when you run them on the gel, even though this two-dimensional gel that you're seeing here might give you multiple spots, but then still it was difficult for scientists to identify. For example, this place or this place, it looks, you can see there are so many proteins which might be present in that dark spot itself. So to overcome this challenge, people came up with another advanced version. Another difficulty with two-dimensional gel electrophoresis was proteins which were very big, that is more than 250 kilodalton in size. They usually find it very difficult to enter into the second SDS page gel properly. They do not move easily into the gel. So separating proteins which are very big in size was very difficult using two-dimensional gel electrophoresis. And the third important disadvantage of 2D gel was variation. That is, every time same sample, same condition, when you try to run it again, every time there was variation. You will never get the same pattern the next time. So that variation created a lot of problem. And less sensitivity, that is lower resolution. Here you can see it is not that easy to analyze this gel. It looks very complicated. So this also scientists found it very difficult. Hence comes our topic, the Dige, which stands for difference gel electrophoresis. Please remember, very important, can be asked as a 2-mark question also. What is Dige? Dige stands for difference gel electrophoresis. It is a very simple two-dimensional gel electrophoresis. It is a type of two-dimensional gel electrophoresis. The only difference being, here we use 3 different types of fluorescent dyes. For example, CY3, CY5 and CY2. CY2 is also sometimes written as CYTM2. CYTM2 I have given in the third point. Okay, so this difference gel electrophoresis is a form of gel electrophoresis where up to 3 different protein samples can be labeled with size matched, charge matched and spectrally resolvable fluorescent dyes. What do you mean by size matched, charge matched and spectrally resolvable fluorescent dyes? That means the CY3, CY5 and CY2, these 3, they are of the same size, they carry the same charge. Hence, when you resolve them on the gel, they will be present at the same spot. But then, the specialty of these 3 dyes is, each of them gives a different color fluorescence. Right? Hence, even though they will be present at the same spot, each one will give a characteristic fluorescent color. Hence, same spot, 3 different proteins, if they are present, all the 3 can be labeled with these 3 dyes. Hence, although all the 3 proteins will be present at the same spot, each one will produce a different color, indicating that at the same spot, 3 different kinds of proteins are present. This was not possible with the normal 2D gel electrophoresis. That is why they came up with an advanced version of normal 2D gel, which is called as difference gel electrophoresis, where we can use different kinds of dyes which are size matched, charge matched. Please underline the word size matched and charge matched. That means whatever dyes you are using, they will be of the same size and will have the same charge. The only difference will be all the dyes that you are using will give different types of fluorescence. Hence, when you know that you want to resolve a particular size protein, you can use these dyes, specific dyes, which will be similar to your protein in size, but then you are not sure that in that spot only one protein is there. So when you use these 3 dyes, you label the proteins and you let them go in. If you find 3 different labels, that indicates presence of 3 different proteins. Let us see how. Here we have taken simple example of these 3 dyes. These are the commonly used dyes, CYTM2, CY3 and CY5. They are mass and charge matched. That means they have the same charge and they have the same size. But they have unique fluorescent properties. Therefore, the labeled proteins will migrate simultaneously on the 2D gel, but still they will produce distinct excitation and emission spectra. They will produce a different color. This means that multiple protein samples which are labeled with different dyes can be co-migrated on the same gel. So on the same gel they can be run, but at the same time they can be easily differentiated from each other based on the type of fluorescence they are emitting. Once the 2D gel electrophoresis is complete, the analysis part you have to use some kind of a fluorescent scanner or a camera system, which will help us in producing the color image. So let us see. You can see here we have 3 different dyes. We have 3 protein sample, protein extract 1, which is labeled with CY3. We have the standard internal protein, which is what we are going to use for comparison. For our example, let us say this blue one, first one, CY2, which is labeled with CY2, that is the protein from a normal person. Let us assume this protein extract 1 is from diseased person 1 and protein extract 2 is from diseased person 2. Right? So first one is labeled with CY2, second is labeled with CY3 and third is labeled with CY5. When you run a normal 2D gel, you can see the gel looks, there's nothing different. Everything looks same. But when you expose them to, when you expose them to the scanner and when you capture the image, fluorescence at different nanometers will give different color. Each dye gives a different color. So this pattern, what you are getting is for the normal protein. This one is from the diseased person and this one is from the normal diseased person 2 and this CY3 is from diseased person 1. So you can see here, this is the same spot. This one is also the same spot. But then just see the variation. Here the protein is present. Here also the protein is present. One, the concentration, you can see the peak formed by each of those protein. Yeah, they are totally different. Also, when you can isolate the protein from the gel itself directly. The protein spot can be picked from there. It can be isolated and it can be studied using different software. So that is what I have explained in the next slide. These are the steps involved. First, we go for protein sample preparation. Then we label the sample with various dyes. Then we do the first dimension separation, which is called as IEF. Then we go for the second dimension separation, which is the normal SDS page. Finally, we go for scanning using Typhoon variable mode imager. This is just an example of a type of a scanner which is used for scanning CY dye. And then we have an analysis software. The name of the software is Dige. Dige. It is a differential analysis software used to analyze the spots. From there we can pick any spot, and that whatever spot has been picked, we can digest it on the gel with trypsin, and after digestion you can pick the spot and go for LCMS. LCMS will help us in analyzing what kind of protein it is. That protein can then be matched with the database available, and you can find out what is the type of protein that you have found. So here is another example. You have 3 different dyes. Sample, when it is run together, you go for first dimension, second dimension, and then you scan it. You can see here, see the difference from the same spot. Different dyes give you different indications that different proteins might be present. So this is what we mean by difference gel electrophoresis. 3 important things which are different from 2D is it uses unique dyes, experimental design is slightly different, and we use decider software which helps in identification of the spot, co-detection of spots, spot volume ratio, normalization, and also you can run ANOVA to find out what kind of sample we have got. A little more information about the fluorescent dyes that we use. Here you can see CY2 excitation peak is at 488 nanometer, whereas emission peak is at 520 nanometer, and it produces a yellow color fluorescence. Similarly, CY3 blue color fluorescence, CY5 red color fluorescence. So this is how each one will get excited, and the emission is at different nanometers. Right? It has a greater advantages. It is pH insensitive. It does not change because of a pH change, and also they are size and charge matched. That means they co-migrate within the gel along with the proteins, and very sensitive. It can identify even protein sample as low as 25 picograms. So here are few examples of images that has been processed. You can see here same spot how there are changes in the image because of presence of one or the other protein. So advantages, whatever limitations we had with the traditional two-dimensional electrophoresis, that can be overcome here using the dye. So with that we complete DIGE or difference gel electrophoresis system. Thank you.